Hey guys, I'm Jazzy Grant, and today we will be discussing human epidermal growth factor receptor 2, or also known as HER2. HER2 is a specialized protein found on breast cancer cells that help control cancer growth. Usually a breast cell has two copies of this HER2 protein. However, when this protein is overly expressed in the body, cells begin to grow uncontrollably and can lead to breast cancer. In fact, women in the United States have a 1 in 8 chance of developing breast cancer during her lifetime. Approximately 15 to 20% of all breast cancers overproduce the growth-promoting protein HER2. Since structure always follows the function in the human body, let's start by taking a look at the length of the HER2 protein. Here you see HER2 amino acid sequence developed using GenPet. The HER2 protein is very long. It consists of 1,255 amino acids. Here you see the theoretical pi value for HER2. The actual pi value is 5.8, while the theoretical pi value is 5.58. This suggests that at a neutral pH, HER2 protein gives a slightly net positive charge. HER2 belongs to the epidermal growth factor receptor, also known as EGFR family, which are receptor tyrosine kinases that help mediate cell proliferation. This EGFR family is a family of 4, which not only include HER2 proteins, but also include HER1, HER3, and HER4 proteins. This makes sense because these kinases are necessary to activate HER2 proteins, which you will soon see. According to secondary prediction tools, as you see here, HER2 is a protein made up of mostly alpha helixes, beta sheets, and random coil turns. You will notice that the structure of HER2 is made up of approximately 28% of alpha helix, 14% of beta sheets, and 58% made up of random coils. This prediction is correct as you see here. Here you notice the alpha helixes, beta sheets, and random coils in this structure. HER family receptors exist on the surface of cells, which contain 3 parts: extracellular domains, which is made up of 4 subunits, transmembrane domain, and an intracellular domain. Let's discuss into further detail the functions of these structures. Firstly, the extracellular domain is made up of subdomains 1 through 4. Subdomain 1, shown here in purple, is constantly in contact with subdomain 3. Permanent interaction between these two subdomains keeps the receptor in an open conformation. Subdomain 2, shown here in purple, is the dimerization domain. This domain enables HER2 to bind with other receptors in the HER family to initiate downstream signaling. Subdomain 3, also shown in purple. Unlike other receptors in the HER family, subdomain 1 on HER2 is constantly in contact with subdomain 3. Permanent interaction between these two subdomains keep the receptor in an open conformation, exposing subdomain 2 and ensuring the receptor is always ready to dimerize. Subdomain 4, shown also here in purple. Its function is still unknown, however it's believed to stabilize and lock the receptor in an open conformation. The second domain is called transmembrane domain. This domain is under investigation, however some suggest that it helps to control receptor activation. Thirdly, the intracellular domain contains a cytoplasmic linker, a tyrosine kinase component, and a tail that phosphorylates and recruits adapter proteins following dimerization. These adapter proteins can then initiate cell proliferation signaling, leading to cell growth. Now that we've discussed each structure's function in HER2, you're probably asking yourself, how does HER2 operate? Well, HER2 does not need a ligand. for activation, unlike the other HER proteins. Thus, HER2 is always ready to dimerize. This is how HER2 is different from all the other HER family proteins. Dimerization then occurs on the intercellular domain of HER2, which results in phosphorylation. This then allows binding of the signaling molecules to these sites. This then leads to many signaling pathways, like the PI3K pathway, which prevents apoptosis of cancer cells, or the MAPK pathway, which leads to cancer cell proliferation. Dysregulation of HER2 will result in uncontrolled proliferation or decreased apoptosis, which both mimic the characteristics of cancer. This makes sense that Cello predicts both extracellular and nuclear movements because the first signal in order to activate HER2 comes from the extracellular domain, and the signal moves from the cytoplasm and continues to travel into the nucleus in order to affect the breast cell. Here you see that HER2 is conserved in domains like mice, rats, and in some mammals. Studies have also shown that HER2 is found in some lung cancers as well. Knowing the structure is extremely important in regards to mutations. There are a total of 4 mutations that occur in HER2. Here you see the structure of HER2. The letters on the right point to specific areas that are altered or mutated in certain naturally occurring or experimentally induced breast cancers. A is the site of somatic mutations found in tumors that come from mice. B is the site of a deletion mutation that naturally occurs in humans. C is the site of the mutation in HER2 where it was initially discovered in a rat experiment. And D, the site of mutations found in rare cases of human lung cancers. As you know now, an overexpression of HER2 proteins will lead to uncontrolled cell growth, which leads to cancer. These cancers, referred to as HER2 positive breast cancer, tend to be higher risk cancers, but certain types of chemotherapy and targeted therapy work well to treat this. Drugs like Herceptin and Perjeta are most commonly used to specifically block HER2 proteins from dimerizing. We can now humbly thank scientific researchers in the 1980s who are hunting for novel oncogenes. Researchers like Schechter and Shi are credited for finding HER2 proteins. With their dedication, perseverance, and focused mindset, we now have a better understanding on how breast cancer evolves and metastasizes. Awareness is key when it comes down to medical illnesses. Be proactive when it comes down to your own body. Self-care is the best care, so care for yourself by first seeing doctors for annual physicals. Get a mammogram screen to help catch breast cancer in its early stages. And most importantly, get tested for HER2 positive breast cancer, so we can help minimize the statistics from 1 in 8 to eliminate. Thank you.